Osmoregulation is essential to aquatic animals for their survival due to the high salt content in their environments. Without osmoregulation, aquatic animals would not be able to excrete excess salt and would perish due to high salt contents in their blood. In this presentation, I will be talking about 3 specific adaptations which some aquatic animals have. These adaptations are medullas in aquatic mammalian kidneys, nasal salt glands in marine birds, and osmoregulation in anadromous fish. Aquatic mammalian kidneys are not like a typical human kidney. Human kidneys are smooth, and as you can see in this diagram, the kidneys of aquatic mammals have many lobes and can vary in size. There are also other differences between a typical mammalian kidney and an aquatic mammalian kidney. A typical kidney, which is shown on the right, does not have an enlarged medulla. The kidneys still function in the same way. Kidneys filter the blood with tiny filtering units called nephrons. They remove toxins from your body and also regulate hormones and other important things in the blood. Kidneys receive blood through the renal artery and then will send it back out through the renal vein when the blood has been filtered. Nephrons run in the cortex and medulla. This is where the blood is filtered and waste is turned into urine. Reabsorption and secretion happen in the tubules, which run through the medulla. Urine is sent through the ureter and then exit the body, and then the blood is sent back into the heart through the renal vein. In aquatic mammalian kidneys, the process is essentially the same. However, they have enlarged medullas to ensure that the aquatic mammal can excrete enough salt and hydrate itself enough to survive in such salty conditions. The enlarged medulla increases the absorption surface so that the kidney can filter more blood and absorb more sodium and chloride ions than a regular mammalian kidney to make more concentrated urine to make up for the salt water the organism is taking in. As I mentioned in the previous slide, the medulla is where the reabsorption and secretion happens. The molecules needed in the blood are reabsorbed as well as water is reabsorbed to hydrate the body. Excess sodium and chloride ions are secreted to regulate the salt content in the blood to maintain homeostasis. The enlarged medulla causes the kidney to have more defined lobes, as shown in the pictures of aquatic mammal kidneys on the right. These kidneys have many small individual ranules, and each ranule is similar to those found in other mammals as well. Selection also acted on these kidneys, and they are selected for because of the genes AQP2, SLC14A2, ACE, and AGT, which are specific for kidneys adapted for saltwater environments. The second adaptation I will be talking about are the nasal salt glands in marine birds. Since marine birds do not have the loops of Henle in their kidneys to filter out the salt content in their blood, they secrete the excess salt through nasal glands. Many marine birds consume their water through eating fish which contain fresh water that will not dehydrate the birds. Some marine birds consume invertebrates, however, so they do not get the same water content out of their food source. This, along with their kidneys, is why they have a need for the nasal salt gland. The nasal salt gland is present in all birds, but it is enlarged in marine birds in particular. In most marine birds, it is located on the top of the head above the eye. The gland is under parasympathetic nerve control, and it is also known as the supraorbital gland or the glandula nasalis supraorbitalis. The gland secretes a solution of sodium chloride, which is more concentrated than seawater. On top of the skull, there are 2 glands with ducts running down beside each other to the nostrils, where the solution can drip out. When the blood increases in salt content, the glands respond through the parasympathetic nerve control. The epithelial cells open up the potassium channels, which allows potassium to flow out of the cell. Then the sodium and chloride ions are moved into the cells via a sodium-potassium chloride transporter. This sudden increase in sodium allows the sodium-potassium ATPase channels to open to remove the excess sodium, which forms an electrical gradient across the chloride ions, which allows the sodium and chloride to enter the gland. The gland then secretes the hypertonic solution of sodium chloride through the nostril. The third example I will be looking at is osmoregulation in anadromous fish. To start off, anadromous fish are a type of fish, such as salmon, which live for part of their lives in freshwater and part in saltwater. For this example, we will be focusing on the salmon species of fish in particular, since they are well known as anadromous fish. In salmon, the adaptive process to seawater is referred to as smoltification. During smoltification, sodium ions and potassium ATPase activities typically increase. Another important part of this mechanism are the mitochondria-rich cells. They are involved in ion excretion and ion absorption. Usually, ion excretory mitochondria-rich cells tend to be larger and are located on gill filaments. There are more of them present in hyperosmotic, saltwater, conditions. Ion uptake mitochondria-rich cells are usually smaller and are on the gill filaments and lamellae, where they decrease in abundance in hyperosmotic conditions. The ion excretory cells increase in saltier conditions in order to excrete a greater amount of salt ions. The ion uptake cells decrease in saltier environments to try and reduce the amount of salt entering the body in the first place. Sodium-potassium chloride co-transporters also play a role in salinity acclimation, where they increase in hyperosmotic conditions. This ensures that the whole process takes place at a faster rate to provide homeostasis for the fish. Different organisms have adapted due to salinity in many different ways, including the ones I have touched on.